Do you know what is the most important step of any genetic study? Well, it is the isolation of genomic DNA. Hello everyone, I am Nandini Jain, a second year BSc Anthropology student from Hansraj College. And today I am here to talk about the topic isolation of genomic DNA from biological matter. Now, before going to understand how we isolate the genomic DNA, first let's understand what do we mean by DNA. DNA, which is deoxyribonucleic acid, it is the genetic material containing all the instructions and the information which are required for be it metabolism, reproduction, heredity, or even the characters of the organism. The DNA holds the blueprint to construct any other cell component or even another cells, such as proteins or RNA molecules. Chemically, the DNA consists of two long strands. These strands are made up of nucleotides. One of these strands is this one, and the other one is this one. These strands have the backbone made up of sugar and the phosphate bonds. These are connected by the ester bonds. Now, these two strands run in opposite directions of each other. That is, they are anti-parallel to each other. Now, let us understand a little about the different types of DNA. There are two types of DNA which are found in eukaryotics. One is the nuclear DNA and the other one is the circular DNA. Nuclear DNA, it is the linear DNA which we find in the nucleus, whereas the circular DNA, it is not found in the nucleus, it is only found in the organelles like such as mitochondria and chloroplast. Now, let's start the topic of the isolation of DNA. So, DNA was first isolated in 1869 by Frederick Mischer. This is the person who first isolated the DNA. He discovered he discovered it and named it as nuclein. Why nuclein? Because it was found in the nuclei of the cell. Now let's understand the genomic DNA. The genomic DNA represents the complete heredity blueprint of an organism, which is which is inside the nucleated cell of an organism. Genomic DNA is isolated in its pure form. To study a DNA, we need to extract it in its pure form, be it from blood, saliva, hair roots, tissues, or we can we can extract it from any biological matter, be it an onion or an human. It is found in everyone and everything, and we can extract it there as well. Now, let us understand what are the different steps of DNA isolation. The first one begins with cell lysis. The next is the inhibition of DNAs, and then we disassociate the nucleoproteins. The fourth step is the removal of contaminants, and the fifth step is precipitation. Now let's understand one by one. Now, before starting the process of DNA isolation, we first need to collect the sample from where we are going to extract the DNA. Now, there are different ways from which we can collect the DNA sample. We can collect the sample from blood or saliva or from plant, muscles, even from the bacteria. Now, the one thing that we need to make sure before we start our process of DNA isolation is the sterility. The DNA can be easily contaminated and would not be extracted in its pure form, and hence we should always make sure that the environment should be sterile. Now, the liquid samples, for example, the blood and saliva, they can be collected in the EDTA tubes, which are these ones, and to prevent clotting. And the hard tissues like plants and muscles, they require grinding. For example, we use mortar and pestle for them. And the bacterias can be treated with enzymes like lysozymes to break down this bacterial cell wall. Now let's move on to the next step, which is the cell lysis. Cell lysis, what do we mean by cell lysis? Cell lysis is the breaking down of the cell. We all know the cell contains of cell membrane, the cytoplasm, proteins, and the DNA, obviously, inside the nucleus. And now to release this DNA, we use a lysis buffer, which contains detergent, and these detergents dissolves the lipid membrane, which is basically our cell membrane, and this releases the DNA into the solution. But when we are doing this experiment in plants, we make sure that we first need to grind it to break the cell wall. Now, the next step is the removal of proteins and contaminants. Proteins are removed using proteinase K and salt solution and phenol chloroform. The centrifugation, then we centrifugate, you can see in this diagram. By centrifugation, the two layers are formed. The upper aqueous layer, which is this one, it contains our DNA, and the lower one, it contains our proteins and debris. Now, one of the final steps is DNA precipitation. Now, DNA is precipitated, but we use cold ethanol or isopropanol, basically alcohol in place of water, because the DNA is insoluble in alcohol. And if we use water, it would be dissolved in it. And after uh, after cold ethanol or isopropanol is added, the DNA then can be spooled out using a glass rod, and it is seen as these white strands. These are the white strands. This is our DNA. Now the DNA is washed and stored. It is washed with 70% ethanol and then dried and then stored at minus 20 degrees Celsius. Now we understand that now the DNA is isolated. But how do we see this DNA? How is it visible to us? Now the step comes is the visualization. How do we see this DNA? We use the agarose gel electrophoresis. The principle of this agarose gel electrophoresis is that it separates the DNA on the basis of its size and charge. That since we know that the DNA is negatively charged, and because it is negatively charged, it will move to the positive electrode. Why? Because we know opposite attracts. The negative is going to move towards the positive, and the positive is going to move towards the negative. So the DNA moves towards the positive electrode. And also, how do we differentiate on the basis of size? The smaller molecules will move faster towards the positive anode, and the larger molecules would be slower. Now we prepare the gel. With gel, these agarose gel. We use 1 gram of agarose powder, which is dissolved in 100 ml of TAE, which is the triacetate EDTA. Now this is microwaved for 1 to 3 minutes, and then it is solidified at the room temperature for about 10 to 15 minutes. Now the DNA, we have to stain. We have now ethidium bromide is added to the gel. The ethidium bromide binds the DNA and allows it to be visualized under ultraviolet lights. We use ultraviolet lights to see the DNA. The DNA appears as these bright orange bands, as we can see here. And let's see a picture of this is how the DNA is represented to us. The smallest one, we can say this is the positive anode, positive electrode, and this is the negative electrode. The smallest DNA, the smallest molecule will move fastest towards the positive end, and the largest one would be slower. And the color of these is the orange, bright orange fluorescent color. Now, there are different methods of genomic DNA isolation. One is the organic method. The one that we discussed right now was the phenol chloroform method. It is a traditional method. We have high purity in this, although it is time consuming and it uses toxic chemicals, but it is the most trusted one. The next come is the column-based method, which is a relatively modern one. It uses silica columns, it is efficient, and it is used in research labs. The last one is the salting out method. It uses high salt concentration and no toxic organic solvents are used, and it is safer and economical. With this, we come to an end of this discussion. I hope you all liked it and understood the concept of isolation of genomic DNA from biological matter. Thank you, and like, share, and subscribe to our college channel. Thank you.